Thank you for joining us. Welcome to day one of the Maxim Group Virtual Growth Conference. My name is Naureen Quibria. I am one of the senior biotech analysts here at Maxim Group. We're very pleased to have Genenta Science with us today. And joining us from Genenta is the CEO, Pierluigi Paracchi, as well as Chief Medical Officer and Head of Development, Dr. Carlo Russo, with us. So thank you both for joining us today, this morning. I guess to get started, also, if any of you in the audience have questions, feel free to just use the button in the web portal. But Pierluigi, Genenta Science is actually new to the market. You IPO'd in last year in mid-December, actually. So, for the audience that isn't familiar with Genenta Science, could you actually talk a little bit about Genenta's core technology, the pipeline, and so forth?
Okay, thank you very much for having us here. And thank you, Naureen, for this question and for this fireside chat. Yes, just let me make just an introduction about Genenta. What we do is that we are developing a cell gene therapy strategy to treat tumors. We are developing a platform, and our technology are based on the capability to express specific anti-tumor payload within the tumor microenvironment, and we are able to control the expression of this protein thanks to a microRNA downregulation. We are focused on solid tumor indications, and the first solid tumor that we are addressing is glioblastoma.
Fantastic. And in terms of your lead drug, which is or lead therapy, interferon, what is it exactly? Can you or Carlo sort of elaborate on that?
Yeah, Naureen, thanks. The basis for the platform that our founders developed is based upon addressing what still remain significant challenges, even in spite of the success of immuno-oncology in the blood cancers. And therefore, the technology platform is to be able to reach solid tumors, without having the very well-known toxicity and tolerability issues that are associated with the majority of the treatment that they're giving IV. And then finally, to address the question that still remain in terms of how durable is the treatment, even when you have a success initially in treating cancer from patients suffering from blood cancers.
Right, and if we go into more detail, it's an ex vivo lentiviral-based therapy, right?
It is.
So, at least recently, there have been a few setbacks, right, with clinical hold and the like. So, can you talk a little bit about your level of confidence, in terms of this lentiviral-based approach to deal with this unmet need in cancer?
Yeah, Naureen, I'm sure that the people listening to this fire chat are aware of the two main gene therapy approaches that are used. The in vivo one with adeno-associated viruses, and then the ex vivo with the lentiviruses. Our platform is ex vivo using the lentiviruses. Now, you're referring to some adverse events that induced the FDA to really put some of the studies in clinical hold. Now, the adverse events are mostly associated with the potential induction of oncogenesis. And this is by the potential disruption either of a silencing gene or activating an oncogene. Now, in our case, it is important to know that the virus is not given to the patients because we modify the autologous cells from the patients in vitro, and then we transplant the cells back into the patient. So The risk of having an in vivo insertion of our virus in different tissues or cells is not present with our approach. In addition, the Genenta is really based on more than 130 patients tested in different clinical trials, not for oncology indications, for rare disease indications, and there are no evidence of any potential mutagenesis associated with the in vivo use of lentivirus transduced cells. So we feel quite comfortable that from a safety and tolerability point of view, our approach is very good so far.
Right, and you actually personally have, you and one of the co-founders, right, Naldini, have personal experience with lentiviral.
Yeah, and this is one of the reasons why I joined Genenta, is because of working with Professor Naldini Tige at the San Raffaele Hospital. We were successful when I was at GlaxoSmithKline in getting the approval of the first ex vivo gene therapy product for children with ADA-SCID, which is a primary immunodeficiency. Now, you know that the ADA-SCID children, the famous bubble kids, our children unfortunately die because of the disease. And I can tell you that one of the most remarkable aspect of the work done with Naldini is that now the children can be cured. Now, we're talking about monogenic disease, very different than cancer, but we have plenty of a technology background platform data that allow us to be quite comfortable in the way we are developing now this technology for cancer indication.
Right. Okay, so yeah, so that's very helpful. And Temferon is tumor agnostic, right? So you can go after many different cancer types, but for your first indication, you're actually targeting glioblastoma, GBM. You know, why go after GBM? Just curious.
Yeah, and it's obviously a very logical, important question, since the GBM remain probably one of the more challenging cancer, even for the immuno-oncology approach. Now, the reason why we feel that GBM, the glioblastoma, is an appropriate target as initial targets for Temferon is based on three different aspects. One, scientifically, there are plenty of publications showing the role of a myeloid cell population, particularly in this cancer, in preventing the immune system to recognize the cancer and have an immune response. From a clinical point of view, the neurosurgeons will tell you that it is remarkable, the massive infiltration of myeloid cells within the tumor. And finally, from a product development point of view, the proposal to introduce for the first time in humans a product never tested in oncology will result in going at the end of the line, meaning after patient fail one treatment after the other. Now, that's particularly a challenge for Temferon, because Temferon, the mechanism of action of Temferon is really based on the ability of the immune system of the patients to recognize the tumor. And therefore, it is important to have a competent immune system. Now, GBM patients, unfortunately, die very, very, very soon after diagnosis, and the cancer is really limited to the brain. And the GBM patient have a competent immune system. And we have now data showing that the immune system of GBM patient can recognize the tumor.
Okay, that's great. So that actually leads me to my next question. You know, you sort of alluded to that. You know, it's been a long haul for GBM patients. You know, there hasn't been any new treatments that have been approved in the last, not therapies, at least last 15 years or so. You know, if we look at small molecules or whatever that are in other modalities that are in development, you know, we might think of them as something more convenient to use. So if you think about Temferon and what you just described, how do you see it fitting in the treatment landscape of GBM?
Yeah, as you know, Naureen, unfortunately for the GBM patients, there is no treatment at the moment that seems to be able to modify the progression of disease. So the treatment available today is radiation and chemotherapy, temozolomide, that unfortunately do not affect the overall survival of those patients. So there are plenty of approaches that now are under development for GBM. The position of our product within the landscape is in a way, I think, unique, because we are using the particular subpopulation of monocytes that seems to be responsible for making the tumor invisible to the immune system. We use them to deliver the treatment, alpha interferon. Now, the data that we have are early in a phase dose ranging study, phase 1, phase 2, and therefore I cannot tell you whether that we already have evidence in terms of modification of disease progression, but in a dose ranging study, we see that the increase of the dose seems to have an effect in terms of the timeline that the patients are undergoing in a MRI assessment of their tumor progression. So I cannot tell you that we have a statistically demonstrate a slowdown in disease, but we have evidence in some of the patients that that may be the case.
Okay, so you just mentioned the dose escalation study that you're currently undergoing with Temferon. So can you just describe, how many cohorts you've actually evaluated to date, how many are left, and so forth, and how many patients?
Yes, so the classical dose ranging study in cell and gene therapy is based on cohorts of small patients, usually 3 patients per cohort. And we have a study that started in 2020 with 3 cohorts with increasing dose and then in January of last year, we analyzed the data. We saw that there was no dose-limiting toxicity, so we amended the protocol. We added 2 additional cohorts for a total of 15 patients. So at the moment, we have treated the 15 patients, and the maximum dose that we use of Temferon was 2 million Temferon cells per kilo per patient. The data so far are showing that the safety is good. There is no limiting dose toxicity, and the patients are responding well to the transplant. We can also see the progeny of cells that derive from the transplanted stem cells up to 2 years post-treatment in the peripheral blood. So we have amended the protocol again to add 2 additional cohorts for a total of 21 patients in a phase 1 part of the study. We will be treating the first patient in a cohort number 6 within weeks, and the maximum dose that we'll be using in a cohort 7, which is the last 3 patients in a phase 1 study, will be 4 million Temferon cells per kilo. So far, I think safety is good, tolerability is good, and the biological activity is measured by the progeny of cells that derive from the transplanted one, and we can measure them even after 2 years of post-transplant.
Oh, wow. Wow, that's impressive. So in terms of recruitment and data update, when you think, say the 16th patient is, have you already enrolled the 16th patient, and how's recruitment going?
Yeah, so we plan to complete enrollment treatment of the last patient in a 21 patient phase 1 part of the study by the end of the year. And based upon the data from the totality of the phase 1 study, we are planning to start a phase 2 study, which will be controlled this time with a synthetic control arm of patients that are matched to a population that will be enrolled in the phase 2 study, and the control will undergo standard of care. This will be a multi-center study still done in Italy.
Gotcha. I was just about to ask, will it be in the US just yet? Okay. So, so you intend to complete the study by the end of the year. So does that mean that you'll have a recommended phase 2 dose by the end of the year, early next year?
Yeah, we will have the recommended dose for the phase 2 study, and we will be, as I said, started the phase 2 study probably end of the second quarter of next year.
Gotcha. And so, with Temperon, obviously the current payload is interferon gamma,
Alpha.
but sorry, alpha. Thank you for reminding me. But you can also plug and play there, right?
Yes.
But in addition to that, you can do combination therapies, right, with Temperon. Can you kind of speak to that? What are your thoughts within GBM itself and in other cancer types? We start with Avastin or any other IOs?
Yes, so it is, Naureen, thank you, because it's important to emphasize the diagnostic nature of our treatment. It is not tumor specific, it's not tumor antigen specific. It is a treatment that will modify the microenvironment of the tumor, allowing the immune system to recognize tumor cells. Therefore, if it works in GBM, it could work in any other solid tumor where the TEMs, this population of monocytes, seems to be responsible for creating tolerance. So our strategy is a standalone strategy, meaning progressing with the GBM indication that we started. But at the same time, our founders, one of the founders, Professor Naldini, is generating very, very interesting data in combination treatment with other immuno-oncology products. There's going to be a paper published very soon, an important paper, where Professor Naldini showed the increase in the efficacy of CAR-T and ICI with the use of Temperon. This is incredibly important because it's a strategy that should be of interest to many other companies that are developing standalone immuno-oncology products, which are being successful but limited in part by some of the challenges I described in the beginning, durability of the response, or the presence of potentially safety associated issues. And Naldini showed that Temperon could be really synergistic with such immuno-oncology approaches in a variety of different tumors.
Great, looking forward to that paper. So, if we're talking about Temperon, just to, the cells generating the therapy itself on the logistical side, how long does it take to take the cells out of the body, to prepare them, create the monocytes and all that?
And again, we are lucky that we're working with teams that have been doing this for many, many years. And as I mentioned before, 120 patients already been treated successfully with this technology. So we are really based on a very, very well tested technology. The product is made by harvesting the stem cells, CD34 cells IV and apheresis, very simple apheresis. Then the CD34 positive stem cell, progenitor stem cells, are given to our manufacturing company, formerly MolMed, now called AGC, which is within the Institute San Raffaele Hospital in Milano. And then the product takes just a few hours of in vitro culturing the stem cells with the lentiviral vector with alpha interferon. Then the product is frozen, which is give us the flexibility to use the product when the patient is ready. In the GBM study, after we harvest the cells from the patient undergoes 6 weeks of radiation, which is a standard of care. And then after 6 weeks, the product is given to the patient IV, simple IV injection. And that's the way the product is made.
That's fantastic. So, you can freeze and thaw the cells. How long, are the cells viable after you've thawed them?
We have a very, very long stability data to the point that we, during a discussion with the FDA, we had a pre-NDA meeting with the FDA. We did ask a specific question: will it be possible if we were to do a study in the US under US IND initially to make the product in Milano and then ship the cells back and forth, the cell from the patient to Milano, the product back to the site? And the answer was yes, and is already done. Was done by other biotech company already. So the stability is sufficiently long to allow us to do that.
Oh, that's great. And you mentioned your manufacturing facility, AGC, did you say?
AGC,
You know,
yeah.
yeah, so, obviously that's an important part of drug development. There have been so many issues with companies, at the tail end when they're about to get approval with that. And with gene therapy, manufacturing is absolutely critical. So, are you going to be relying on them in the future? What are your thoughts on skills?
Yeah, very good question, Naureen. If you remember, a few years ago, most of the biotech companies that were interested in doing cell gene therapy, they want to build their own manufacturing facility from the get-go. Even before having any evidence of potential success. The reason why, because at that time there was no market for cell gene therapy, and therefore there was really not much of an offer from CMOs to do cell and gene therapy. Now we are really in a very different places, and you know that there are plenty of CMOs that can provide you with cell and gene therapy services. Therefore, I feel quite confident that yes, we can work with AGC as it is, but in the future, if we were to expanding, AGC also is in the United States, but we're also talking with different vendors to define what will be the best for us in terms of manufacturing the product, not only in Europe, but also in the US. So I don't think the manufacturing will be for us a significant challenge. We will be able to address when the time is right.
Okay, got it. So, pivoting just slightly, we're almost up on time. So what's the next indication for Temperon that you're looking at?
As I said in the beginning, the platform is agnostic. Mm-hmm. And therefore, we had assessed a variety of different solid tumors. And the criteria that we use are basically, are the tumors showing the presence of this population of TEMs? Yes. Are the tumors inhibiting the immune system response? In other words, there are evidence of tolerance? Yes. Are the tumor accessible? That will be an important aspect. And we have a variety of different tumors that go from liver cancer, adenocarcinoma, for example, to other cancer, breast cancer, and Professor Naldini has already published many papers on animal models of cancer where TEMFR is being tested and used. So we are assessing the best second solid indication, and we plan to do this in 2023 to have a decision on what could be the second solid tumor indication.
Okay, great. And so, okay, we're very close to them. So Genenta obviously isn't merely a one therapy company. You actually have a whole pipeline and several platforms. Can you talk about the other assets in the pipeline? Do you have any ongoing preclinical work?
Yeah, and Professor Naldini is working on several different projects, including different payloads that are not disclosed at the moment, and we have exclusive right options on those. We is also working on potentially switching on and off the product after it's been given to the patients, and is also, as we discussed already, working in combination treatment with different cytokines payloads, as well as a potential switch on, switch off second generation products. So the pipeline is quite large from that point of view. And we are very comfortable that after the proof of concept is established in humans with Temperon, we'll be able to come up with new interesting proposals in terms of combination treatment, and perhaps in certain tumor, a new payload to be used.
Right, so that's fantastic. So, okay, one final question for Pierluigi, very basic, what's the current cash position, runway going forward, and perhaps can you leave us with some thoughts about where investors should focus for the next 6 to 12 months for Genenta?
Thank you for that, especially in this market condition, the cash position is important. Thanks to the proceeds of the IPO, and for us, the December IPOs was a 20% upsides IPO. We have enough cash until the end of 2024. So we can proceed with our program, we can move on with our strategy at the clinical stage, and this is something that can be very important for us and for the company and for the future. About the next step, Carlo already mentioned a lot about the data and what we are going to present in the near future, especially the end of the phase 1/2, the dose range study at the end of this year, and then on the first part of 2023, we will start the phase 2 in GBM.
Fantastic. Thank you both for joining us, and thank you to the audience. We'll look forward to following Genenta. Have a great day.
Naureen, thank you so much.
Thank you.